Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
For decades, we really thought genes worked in, well, one primary way. Making proteins. Blueprints for everything, right?
Right. The central dogma, pretty much.
Yeah.
DNA to RNA to protein.
But what if that wasn't the whole picture? What if a tiny piece of RNA, something non-coding, was actually pulling the strings, running a whole developmental clock?
That's exactly what this deep dive is about. A discovery that really flipped that assumption on its head.
Welcome to the deep dive. We sift through fascinating sources, pull out the key stuff, basically give you a shortcut to getting informed.
Yep. Less reading, more understanding.
Today, we're digging into a truly game-changing paper from 1993. Lee, Feinbaum, and Ambrose. It's where microRNAs, though not yet named that, first stepped into the light.
We've got the original paper, the C. elegans heterochronic gene LIN4, encodes small RNAs with antisense complementarity to LIN14. Quite a title.
Yeah, academic titles.
And we've also got some analysis looking at just how innovative it was. It's a classic, really redefined gene regulation. Showed us a whole new layer.
Okay, so let's set the scene. You've got this tiny worm, C. elegans, a biologist's favorite model organism for a reason.
Absolutely. Transparent, simple, develops like clockwork.
And it goes through these really precise larval stages, L1, L2, L3, L4. Scientists knew certain genes controlled this timing. They called them heterochronic genes.
LIN4 and LIN14 were the big players that were focused on here. They knew these genes were key for the timing.
But how they worked, how they controlled that clock, that was the mystery. Like, what was the actual molecular signal passing between them?
It was a black box. They had strong genetic clues, though. Classic work showed if you lose LIN4, the worm just keeps repeating the early larval stages. It gets stuck.
Like a developmental stutter.
Exactly. And if LIN14 was overactive, you saw a similar thing, a slowdown. It couldn't progress properly. So the genetics screamed, LIN4 must somehow shut down LIN14.
Okay, LIN4 inhibits LIN14. Makes sense. But then came the puzzle.
Right. The LIN14 protein, the thing doing the work, its levels, dropped sharply after that first larval stage L1, just like you'd expect if it was being shut down.
But, and this was the kicker, the LIN14 messenger RNA, the blueprint for the protein, its level stayed constant, didn't drop at all.
Whoa! Okay, that doesn't fit the simple picture. If you're stopping the protein, you'd think you'd stop the message too.
Precisely. That strongly suggested that control wasn't happening at the DNA to RNA step, transcription. It had to be after that, post-transcriptional.
And they suspected a specific part of the mRNA.
Yeah. Previous work pointed to the very end of the LIN14 mRNA molecule, a region called the 3' untranslated region, the 3-UTR, sort of a regulatory tail.
So the big question was hanging there. What was this mysterious thing made by LIN4 that was latching onto the LIN14 3-UTR and telling it, okay, stop making protein now? What was the messenger?
That's the million-dollar question they set out to answer.
And to even start, they first had to actually find the LIN4 gene itself.
Which wasn't trivial back then.
No, definitely not. This was early 90s genetics. They used techniques like chromosomal walking.
Which is kind of like inching your way along the chromosome using known landmarks.
Pretty much. And RFLP mapping, using natural variations in DNA sequence as markers to pinpoint the gene's location. Think of it like genetic triangulation. Really painstaking work on chromosome 2 of the worm.
Molecular detective work?
Totally. Once they had a region, they used something called transformation rescue. This is clever.
Okay, what's that?
You take worms that have a broken LIN4 gene, the one stuck in development, and you inject them with different small pieces of DNA from the region you suspect contains the working gene.
And you see which piece fixes the problem.
Exactly. Which piece rescues the normal development? They found that a specific mutant, LIN4, E912-4, had a chunk of DNA missing, which they showed using a southern blot. That's figure 2.
A southern blot lets you see specific DNA pieces, right? Like a DNA fingerprint.
Yep, and it showed that deletion clearly. But the really key finding, highlighted in figure 1, was that they could rescue the LIN4 function with an incredibly tiny piece of DNA. just 693 base pairs.
693, that's really small for a gene controlling something so fundamental.
Way smaller than you'd expect for a typical protein coding gene. That was a big clue. Red flag number 1, maybe.
And this leads right into the next big surprise because everyone, absolutely everyone, thought genes made proteins. That was the rule.
It was the dogma. So naturally, the next step was find the protein Lin4 makes. Seemed obvious.
Not.
But this is where things got really interesting and where Lee, Feinbaum, and Ambrose really showed their scientific nerve. They looked for open reading frames, ORFs.
Those are the sequences that actually code for amino acids, the building blocks of proteins?
Right. They looked for ORFs in the Lin4 sequence from C. elegans and compared it to the same gene region in 3 other related worm species. Looking for something conserved, something important enough to be kept across evolution. That's figure 3A.
Makes sense. If it's an important protein, the sequence should be similar.
You'd think so. But figure 3B shows the shocking result. Absolutely no conserved ORFs. Nothing that looked like a real protein coding sequence shared between these worms.
No conserved protein blueprint.
None. And they didn't stop there. They deliberately introduced mutations, frame shifts, stop codons into the sequences that might have been ORFs, even if they weren't conserved.
Mutations that should totally wreck a protein if it was being made.
Absolutely obliterates its function.
Yeah.
And nothing happened. The Lin4 gene still worked perfectly fine.
Wow. So strong evidence against a protein.
Really strong.
Yeah.
It basically screamed, Lin4 does not work by making a protein, which back then was like saying a car engine runs without pistons. It just didn't compute easily.
So if it's not a protein, what on earth is it? They had to hunt for something else, something unexpected.
And something probably small, given that tiny 603 base pair fragment. This required serious technical skill. You're looking for something potentially rare, definitely small, maybe unstable.
Not an easy target.
Not at all. They used a whole battery of sensitive techniques. Northern blots, RNase protection, primer extension, S1 mapping.
Yeah.
These are all ways to detect and precisely map RNA molecules. You can see the probes they designed in figure 4.
Okay, so RNA hunting, what did they find?
This was the breakthrough moment. They found 2 small RNAs made by Lin4, a longer one, Lin4L, about 61 nucleotides.
Still tiny.
Very tiny. And an even shorter one, Lin4S. Only about 22 nucleotides long, but much more abundant. You see them clearly in figure 5.
22 nucleotides, that's minuscule.
It really is. And they confirmed these weren't just random bits of RNA junk. They were absent in the Lin4 mutant worms.
The ones with the broken clog.
Right. And they reappeared when they rescued the worms with the working Lin4 DNA fragment. Figures 6 and 7 show the incredible detail they went into, mapping the exact start and end points of these tiny RNAs.
So the gene wasn't making a protein, it was making these tiny RNAs.
Exactly. And they nailed it down even further. They found a mutant worm, man767, where just a single DNA letter changed a C to a T at position 517, shown back in figure 3A, and confirmed in figure 7, completely killed Lin4 function.
And that single change was right within the sequence of these small RNAs.
Smack bang in the middle of them. That directly linked the small RNAs to the gene's function. It was proof. These tiny RNAs are the active product of Lin4.
Okay, so they found the messenger, tiny RNAs. Now, how did they connect them to Lin14? How do they work?
This is the final piece, the aha moment. They looked at the sequence of their newly found Lin4 small RNAs and compared it to the sequence of the Lin14 mRNA, specifically that 3UTR region everyone suspected was important.
The regulatory tail.
And what they found was stunning. The Lin4 RNAs contained sequences that were complementary, like a mirror image, or the other half of a zipper to specific sequences within the Lin14 3UTR.
So they could stick together, RNA binding to RNA.
Precisely, and not just complementary to one spot. There were 7 imperfectly repeated sites in the Lin14 3UTR that the Lin4 RNA could potentially bind to. This concept is shown in figures 8 and 9. It was beautiful.
And these binding sites were in the exact region that other mutations, the Lin14 gain-of-function ones, had pointed to.
The very same region. All the genetic and biochemical evidence suddenly clicked into place.
So the model became, the Lin4 gene makes these small RNAs.
The small RNAs then physically bind to the 3UTR of the Lin14 mRNA.
Through this complementary sequence pairing.
And that binding somehow stops the Lin14 mRNA from being translated into LN14 protein.
Which explains the paradox. mRNA stays high, protein goes down, the RNA isn't being destroyed, it's just being silenced.
Exactly. Translationally repressed. It was a totally new mechanism for gene regulation by an endogenous molecule, RNA regulating RNA through direct binding. Revolutionary stuff. No one had really proposed this for how developmental timing genes worked.
Just incredible. So, stepping back, what's the real significance here? This wasn't just figuring out one worm pathway, was it?
Oh, far from it. This was a landmark, a
Yeah.
paradigm shift. They zeroed in on this specific gap. Protein-centric models couldn't explain that Lin-14 mRNA protein paradox.
Right.
So they reasoned, logically, if Lin-4 acts via the Lin-14 3-UTR array, maybe this product isn't a protein. Maybe it's something different, a non-canonical regulator.
They were willing to think outside the protein box.
Totally.
Mm-hmm.
And by doing that, they didn't just solve the worm developmental clock puzzle, they fundamentally expanded what we considered a gene to be. It could be something that made a functional RNA, not just a protein.
It's huge.
And think about their process, that design, validation, feedback loop, positional cloning, rescue experiments to get the gene,
Meticulous work.
and rigorously disproving the protein hypothesis with cross-species comparison and mutagenesis.
Showing what it wasn't.
Then the incredibly tough biochemistry to actually find those tiny RNA.
Which nobody was really looking for.
And finally, that antisense logic, finding the complementarity, proposing the RNA-RNA binding model. This was years before microRNA was even a word, long before we understood the RNAs complex machinery involved.
It really sets a template for discovery. And you mentioned the environment, the collaboration.
Yeah, that's crucial too. Being at Harvard, having the elegans genetic toolkit available was obviously key. But also that open exchange with Gary Ruvkun's lab, who were working on related problems.
They acknowledged each other's work.
They did, citing unpublished data, sharing insights. That kind of cooperative spirit, rather than intense competition, probably sped up the whole process significantly. Science at its best, really.
And the individuals involved, Lee and Feinbaum, the first authors,
Showed incredible technical nerve, as one analysis put it, pushing those RNA detection methods to their limits when the easy path was looking for a protein, and Victor Ambros providing that genetic framework and, backing those bold, dogma-defying experiments.
It takes courage to go against the grain like that.
It really does, especially when the grain is the central dogma of molecular biology.
So thinking about the breakthrough positioning, how did this discovery change things moving forward?
Well, theoretically, it was massive. It launched the whole idea that tiny endogenous RNAs could act as timing regulators, as gene silencers, by binding to 3-UTRs, a completely new layer of control.
The birth of the microRNA field, essentially.
Pretty much. Technologically, it gave people a roadmap. Find minimal functional DNA, map small RNAs, look for complementarity and potential targets. Others use this exact playbook to find LET7, another super important microRNA, and then countless others.
So it wasn't just a one-off finding, it was generalizable.
Highly generalizable, it turned out. And in terms of application, while maybe not immediate 93, the implications were vast. We now know mRNAs are critical in development, cancer, stem cells, pretty much all over biology and medicine. This paper laid the conceptual groundwork.
Were there limitations at the time, things they didn't figure out?
Oh, sure, like any pioneering study. They didn't identify the protein machinery involved, what we now know as the RNAs complex, with argonaute proteins and so on. That came later.
Okay.
And they couldn't know for sure in 1993 just how widespread this mechanism would be. Was it just a weird worm thing or something bigger?
Which we now know it definitely was.
Absolutely. It turned out to be universal across animals. Even plants have related pathways. So yeah, despite those initial unknowns, it was undeniably a paradigm setter. It cracked open a whole new field.
So this deep dive really shows how tackling a fundamental puzzle in a humble worm and being willing to challenge core assumptions led to this huge discovery. Tiny non-coding RNAs as master regulators.
It's a fantastic story, finding massive significance in the minuscule. And a reminder that the biggest breakthroughs often come when you look where others aren't, or see something others dismiss because it doesn't fit the current model.
It really makes you wonder, doesn't it? Which brings us to a question for you, the listener.
How many other basic biological processes might be controlled by mechanisms we haven't even thought of yet, simply because we're locked into our current ways of thinking?
What are we missing just because it seems counterintuitive right now? This paper is a powerful lesson in the value of following the data, no matter how strange it seems.
Makes you think about what other non-coding parts of the genome might be up to, doesn't it?
Definitely. What stood out most to you about this journey into the birth of microRNAs? We hope it gives you a fresh angle on how life manages its incredibly complex timing. Until next time, keep diving deep.